Ladies and gentlemen, welcome to my molecular biology seminar. My name is Isabella Peláez, and today I'm gonna talk to you about the article Aurora kinase inhibitor MLN A237 suppresses pancreatic cancer growth. Let's begin. Okay, so pancreatic ductal adenocarcinoma, known as pancreatic cancer, is a lethal disease with a 5-year survival rate of 9%. And even though there are treatments that exist for this cancer, as we can see, they aren't quite effective. And that's why we can say that there are limited options of appropriate chemotherapy drugs. However, recently scientists have found out that there is an enzyme known as Aurora kinase ARCA that plays an essential role in progression of cellular division, and it is overexpressed in this type of cancer, associated with high tumor grade and increased risk of cancer, in addition to chemoresistance. On the other hand, we have MLN A237, which is an pyrimidobenzazepine alisertib. Pharmacologically, it is a compound with benzene fused to azepine. And basically, this is a gene specific for ARCA inhibitor, which is in phase 3 of evaluation, promising preclinical data and anti-cancer activity through inhibition of proliferation and migration. So basically, it inhibits tumor growth. Then we have the objective of the study, which is to evaluate how Aurora kinase inhibitor MLN A237 suppresses pancreatic cancer growth. Moreover, we have the methods of these experiment trials, which were cell lines and cell culture, which is basically a process by which cells are grown in a favorable artificial environment. And in this article, they were used to determine if MLN A237 could get affected in pancreatic cancer cell proliferation, tested in these different type of cell lines, which were ASPC1, BXPC3, MIAPACA2, and PNC1. We also have transwall assay and Western blotting, which consists in the reaction between antigen and antibody, and it is based on these two principles, which were electrophoresis and ELISA. And in the article, they were used to evaluate the levels of ARCA, which was the antibody, in a normal pancreas and in cancer tissue. In addition, we have immunohistochemistry staining, which detects antigens from a cell tissue. And in the article, they were used to confirm the increased expression of ARCA in cancer tissue from human PDAC and pancreatic cancer animal model, which leads us to our next point, which is mouse orthopathic and genetic pancreatic cancer models. As we can see, we have orthopathic pancreatic cancer models with tested with a 6-week-old male nude mice, and genetic pancreatic cancer models tested with a P53 LSL PDX cream mice. Both of them were used with different types of cells, but the gene that was common for both was the MLN A237, which was the inhibitor of ARCA. For the results, we have chart A, tested with Western blotting, which analyzes the ARCA comparison in normal tissue and how elevated we can see it in pancreatic cancer. Then we have chart B with the results that come from immunohistochemistry, and it shows how in humans and mouse tissue the ARCA can be expressed in pancreatic cancer. Moreover, we have chart D results in which we can see how cells were treated with MLN A237, except for this first square, but for all of the rest, we can see that there were two null positive cells, and apoptosis could get confirmed. In chart B, we can see how MLN A237 is increased in each cell line, especially in caspases, which leads us to our theory, to confirm our theory that MLN A237 is influenced with apoptosis. And then we have how in chart D, how MLN A237 is expressed and increased in each cell lines in these different samples. To continue with the discussions, we can see that the authors C, Neil, and E, the three of them contributed with their opinions to the central idea of this experiment and are in favor of the central idea, which was inhibiting the aura kinase that was overexpressed in a pancreatic cancer. To conclude, thanks to the variety of molecular biology tools that exist right now, they are the ones who allows us to understand how a disease such as cancer could get developed, and if we can understand how cancer comes and develops, we can understand and we can kind of work with the same tools to know a possible solution of how we can contribute to this type of cancer. So in this article, we can see that scientists with these molecular biology tools could find a solution that could suppress the function of the enzyme, which was the aurora kinase. And we also see that these tools, these biology molecular tools, are necessary for scientists to develop their ideas and to confirm and have evidence if the experiment they are testing could work or not. And the only way is by testing with these tools. So thank you very much, and this is all for today.